Dear students, welcome back to another session of biology classes presented by Chaitra, lecturer at Shakthi School and PU College, Shakti Nagara. Last part we had discussed about the important questions from Heredity and Evolution chapter. We had gone through the mono-hybrid cross and we had stopped there. Today we shall continue and complete the chapter in this part. I hope you all remember what was done in the last part. Quickly recall and let us get ready for this session. Today we shall begin with the dihybrid cross, students. So what is dihybrid cross? If you remember, last class we had explained mono-hybrid cross, the cross between parents differing in one character only. Whereas dihybrid cross is a cross between parents differing in two characters. So we have two parents, students, who are exactly similar to each other, but they are differing in two characters, or they have two contrasting characters. So we shall see what are the characters used by the father of genetics, that is Gregor Mendel. He used round and yellow seeds, and for the second parent he used wrinkled and green seeds. So when I say round and yellow seeds, it means that the parent is producing round and yellow seeds, students, and the other parent is producing wrinkled green seeds. Please do not get confused. It is the seeds produced by the plants, that is round yellow and wrinkled green. Since these are the parents that Mendel takes for his experiment, we shall call this as the P1 generation. If you remember, we are always supposed to start with P1 generation for any cross. Next, we write the genes or the genotype of the P1 generation. Now, if you remember, the round is represented as capital R, and yellow is represented as capital Y. So, round, we write it as capital R, and yellow, capital Y. Following the same pattern for wrinkled, we write it as small r, and for green, we write it as small y. Once we have written the genes, what is the next step? We are supposed to write the gametes. Now, what are gametes? How do we write them? Quickly recall. So, gametes are always supposed to be written within circles. So, we have, the rule is, if you remember, inside one gamete, we can write only one letter of one kind. So, since there are two R's here and I cannot write both of them in the same circle, I will draw two circles and I will write two R's. Same thing, we have two Y's. Inside one circle, I'll write one Y, another one, I'll write the other one. Following the same pattern, we have small r, small y and small r, small y. Next, what did we do? Pollination. Mendel pollinates the flowers, correct? So, pollination takes place, and during pollination, we know that either this gamete can meet this one, this or this, and the same thing. So, we have four options, students. First gamete meeting the first gamete, that is capital R, capital Y, small y. So, we have capital R, capital Y, small y. Second, we have capital R, capital Y, small y. Third, also we have capital R, capital Y, small y. And the last, we have capital R, capital Y, small y. After this, what do we write? Next, we have stopped till here. Mendel does this experiment in his garden, and as a result, he finds that all the plants that he obtained after this pollination, when he grew those seeds, he got the plants and all the plants started producing round yellow seeds. So this was round yellow, students. This was round yellow, round yellow. So we have 4 round yellow seeds in the F1 generation, that is the filial one or the first generation, we have only round yellow seeds. Next, we shall proceed quickly with the F2 generation. How do we obtain F2 generation plants? So, the P2 generation. If you remember, Mendel selects any two plants from the F1 generation as parents. So, what were the F1 generation plants? They were round yellow. All the plants were round yellow. They were producing round yellow seeds. So, round yellow seeds. What was the genotype, students? It was capital R, capital Y, small y. Capital R, capital Y, small y. Next, we will call this as the P2 generation, and these are the genotypes. Next, we are supposed to write the gametes. Gametes. Now, here we have lot of options, correct? The rule was inside one circle you can write only one letter of one type. So, we have to write this together, this. So, I'll explain it better. So, we have 4 options for one plant and another 4 options for second plant. So, total we have 16 options, students. Lot to write. That's why to make things simpler and easier, we draw something called as the Punnett square. So, what does the Punnett square look like, students? It has 5 rows and 5 columns. As you can see here, it's a big box. So, when you draw it for the exam, make sure that you're drawing it neatly, bigger the better. So, always draw it neatly and write everything inside the box very neatly, even if it has not been mentioned in your textbook. I'll tell you what to do with this. So, we shall first start filling the Punnett square. The first box is always supposed to be filled as male and female. So, it means this row represents the gametes from the male or the father, and this column represents the gametes from the female or the mother. Once you have filled this, the next step is to fill the gametes. So, as you remember, gametes are always supposed to be written inside the circles, correct? So, we shall draw the 4 gametes first. Once you have finished drawing the gametes, what do you do next? Fill in the genes or the genotypes. So, how do we do this? Very easy, students. First, you write, you know the parental genotype or the genes of the parents, correct? Capital R, capital Y, small y. Once you know this, first box you will have both capital. Second box, one will be capital, other will be small. Third box, one will be small, other will be capital, and the last, both of them will be small. Very easy. So, first box, both are capital, R and Y. You remember the rule, inside one circle, only one letter of one kind. So, I cannot write 2 R's together. That's why I am writing R and Y. Both are capital. Second box, one will be capital, other one will be small. Third box, one will be small, second one will be capital, and fourth box, both of them will be small. Same thing we are going to copy it down here. Capital R, capital Y, capital R, small y, small r, capital Y, small r and small y. Once we are done with this, we start filling in the genotypes. So, first box is this. How do we fill this? First box from here and first box from here. What do we have? Capital R, capital R, capital Y, capital Y. Second box is nothing but this and this. So, we have capital R, capital Y, small y. Third box is this and this. We have capital R, capital Y, capital Y. And the fourth box is this one and this one. Capital R, capital Y, small y. Same way we shall fill all the boxes, students. So, the first box will be this and this. second one this and this, third one this and this, fourth one second and fourth box. So very easy. R, R, Y, Y, capital R, small y, capital R, small r, capital Y, small y, capital R, small y, small y. Next we have capital R, small r, capital Y, capital R, small r, capital Y, small y. Very easy. Next we have small r, capital Y, and last we have small r, capital Y, small y. The last row, students, we have capital R, capital Y, capital R, small y, This box is nothing but small r, capital Y, small r, small y, small y. Once we have filled in all the genotypes, the next step is to fill the phenotypes. How do we do it? We'll take all the round yellow first. We'll see which are round yellow. How do we find out, students? Remember, capital R stands for round, and small r stands for wrinkled. So wherever we have capital R, we'll fill it as round, and wherever we have small r, we'll fill it as wrinkled. So we have the first here, capital R. So this is going to be round. Here again we have round. Here we have capital R, small r. If you remember the P2 generation, we had capital R, small r. What was that? It was round. So this is also going to be round. And this one is also round. Here again capital R stands for round. Here also we have round, and round. Next we have capital R, small r. Again we have round. This is also round. Small r is wrinkled, correct? So we'll leave it like that. We'll fill it as wrinkled. So wrinkled, and again small r is wrinkled. So we'll fill it as wrinkled. Here we have capital R, small r, which is round. This is also round. Small r is wrinkled, and again small r is wrinkled. Once you are done with all this, we will now fill in the Y, or the yellow color or the green color seeds. So again we'll quickly remember the genotypes. Capital Y stands for yellow. Capital Y, small y stands for yellow. Only small y stands for green. Very important, we need to remember this. So we'll quickly fill in all the yellow ones first. So capital Y is yellow. Capital Y, small y is again yellow. This is also yellow. Yellow seeds. This is also yellow. Small y is green. We'll leave it like that. This is yellow. This is green. This is yellow. Capital Y. Next, capital Y, small y is yellow. Capital Y is yellow. And we have capital Y, small y, again yellow. Only in this row we will have two greens. So this is yellow. This will be green because it's small y. Capital Y, small y will be yellow. And this is going to be green. Now we'll quickly fill in the green boxes. We have small y, which is nothing but green. Next we have small y here, which is again green. Then we have here, which is green, and here we have green. So after this box has been filled, what is the next step? We start counting them. We see how many round yellow we have. So we'll quickly count. Round yellow, 1, 2, 3, 4, 5, 6, 7, 8 and 9. So we have 9 round yellow seeds. Students will quickly write it down. 9 round yellow seeds. So 9 round yellow. Next we have wrinkled green. How many wrinkled green do we have? Let us count. Wrinkled green, 1. Only 1. So wrinkled green is 1. Next, what are the options, students? Round yellow is done. Next will be round green. So quickly, round green 1, round green 2, round green 3. So we have 3 round green seeds. Round green is 3. Next, we are left with wrinkled yellow. So wrinkled yellow 1 here, 2 and 3. We have 3 wrinkled yellow seeds. So what is the ratio, students? Round yellow is to round green is to wrinkled yellow as to wrinkled green. We have 9:3 is to 3 is to 1. Very, very important, students. The phenotypic ratio is 9:3 is to 3 is to 1. If you have done the cross properly, however you do it, you will always get this ratio. If you interchange the gametes, no worries. You simply write down the table, fill everything. At the end, you just count. If you have round yellow 9, wrinkled green 1, round green and wrinkled yellow 3, then you are good to go. Your table is absolutely right, your ratio is perfect, and you have the dihybrid cross. So please remember the phenotypic ratio, students. You will not be expected to remember the genotypic ratio at this level, so you need not worry about it, but phenotypic ratio is a must. With that, we have finished the dihybrid cross. We have finished both the experiments of Mendel. We shall now see what is the conclusion of his experiments. As a conclusion, he has given 3 laws, students. So we'll quickly go through the laws, very important. So we have the 3 laws. First one is called as the law of dominance. The law of dominance. This is the first law of genetics, students, the law of dominance. What does it say? According to the law of dominance, Mendel has given 3 statements. Number 1, all the characters are controlled by genes. So first he says, characters are controlled by genes. Very important. This we have already seen in the last class, correct? That was characters are controlled by genes. I had given an example as to how to say that characters are controlled by genes. So quickly recall that. Second, he says that genes always occur in pairs. Very important. What does it mean, students? Genes as in capital T, capital T. You never write it singularly. If you have noticed, it's always together. Capital R, you write it 2 times. Capital Y, again 2 times. So these are genes and we always write it twice because in our body, the genes always occur twice, meaning we have 2 sets of chromosomes. So we have 2 pairs of genes. Third one is we have something called as the dominant trait and the recessive trait. Now this is very important and can be asked as a separate question. What is dominant trait and what is a recessive trait? When you have a pair of dissimilar characters, for example, if I have capital R, this is similar character. Small r, again it's similar. But when I have Capital R, small r, they are dissimilar, students. So when I have a pair of dissimilar genes, one gene is dominant and the other gene is recessive. So I'll repeat, when I have a pair of dissimilar genes, one gene is dominant and the other gene is recessive. The dominant gene is represented in capital letter. That's why capital T as for tall, capital R as for round, capital Y as for yellow. All these are dominant characters, students. They are written in capital letters. The other gene that we write it in the small letter is nothing but the recessive gene. We have dwarf, small t, we have green, small y, and we have r, small r, that is nothing but wrinkled. So please remember the dominant characters and the recessive characters. They are always written in capital and small letters respectively. Now, what are dominant and recessive trait? Naturally, the question comes to our mind. What Mendel says is, when we have a pair of dissimilar character, one character can express itself, the other character cannot. The character that can express itself is called as the dominant character and the other character is called as the recessive character. I'll repeat, the character that expresses itself means in the body you can see that character. That character we call it as the dominant and the other one we call it as the recessive character. So I hope we are very clear with the 3 points under law of dominance. All the 3 points are must, students, for your answer. First one, characters are controlled by genes. Genes always occur in pairs, and when we have a pair of dissimilar genes or dissimilar characters, one will be dominant and the other is going to be recessive. With that, we shall end the first law of genetics, that is the law of dominance. Coming to the next law, that is called as the law of independent assortment. And we also have the law of segregation. These laws go hand in hand, students. So we have the law of segregation and the law of independent assortment. What is it? We'll start with the process called as segregation. Now if you remember, when you were writing the cross, we split the 2 letters and I stated a rule. Inside one circle, only one letter of one kind. Correct? So when we had 2 capital T's together, we separated them and we wrote it as capital T, capital T. This is nothing but segregation happening, students. Segregation is nothing but separation. So the 2 T's are separating here. That is why these are nothing but genes and the circles are gametes. So the genes are separating in such a way that only one gene is entering one gamete. That is the law of segregation. The genes separate in such a way that only one gene enters one gamete. If you write so much, it should be good enough. So the genes split or separate. Segregate is nothing but split or separate such that only one gene enters one gamete. Very important, students. One gene enters one gamete. Only one sentence from this law should be good enough. You need not learn it in detail, but the statement you should know. Coming to the third law, that is the independent assortment. Again, this is very similar to the segregation law. What does independent assortment say? Let us quickly recall the dihybrid cross. We have capital R and small r, small r. Here we had capital Y, small y, small y. This was what dihybrid cross was about, correct? Now, we have 2R and 2 Ys. They will split. How do they split? Quickly recall. We wrote it like this. The R split and only one gamete entered, our segregation. Secondly, Y also split and it entered only one gamete each, meaning both of them are segregating, but they are not dependent on each other. I'll repeat, this is also segregating, this is also segregating, but it is not dependent on each other. So the law of independent assortment says when we have two pairs of characters, the splitting of one pair of character is independent of the other pair. That is independent assortment, students. The law of independent assortment. That is nothing but splitting of one pair of characters is not dependent or is independent of the splitting of another pair. With that, we have completed all the three laws from Mendel. The first law, second and the third. Dominance, segregation and independent assortment respectively. We shall next proceed to the second half of the chapter, that is evolution. From this, we shall only touch upon very important questions. One obvious important question is what is evolution, and second one is what is speciation, and third one is what are the two different types of traits. So we'll quickly go through the first question. What is evolution, students? Evolution is nothing but it is a biological process in which slow and steady changes take place in a given population. So if I have a population, let us say population of elephants. Now, these elephants over millions and millions of years might undergo slow changes in their body form, and that is nothing but evolution. So evolution is a very slow process. It takes millions of years to happen, and it happens in a given population. These 3 points are must in your answer, students. With that, we'll finish evolution and quickly go to what is speciation. Again, commonly asked question. From the evolution part of the chapter, speciation is the most important one. What is speciation? In the name itself, we have it. Speciation is nothing but process of formation of new species. So new species are formed, students. So the process of formation of new species is nothing but speciation. Under this, you are supposed to explain what are the causes. We have 3 causes. First one is the geographical isolation. Geographical isolation. Second one is gene flow, which you might be very familiar with. And third one is the genetic drift. So we have 3 reasons for the formation of new species. First one is the geographical isolation, second one is the gene flow, and third one is the genetic drift. Quickly explain why these are called as the factors for formation of new species. You need not study them in detail, but in case you have some doubts, we'll quickly go through it. Geographical isolation. Remember the example given in your textbook about the beetles. We had 2 types of beetles, maybe green and red beetles. And one day, these 2 beetles were separated by a river. You remember that example, and you will see that because there was a river, it led to speciation. So, geographical isolation means 2 areas, geographies are separated from each other by some physical barrier. That we call it as geographical isolation. Maybe river, maybe island, all these are examples for this. Coming to gene flow and genetic drift. What is gene flow, students? Due to some reason, when a gene is flowing from one population to another, then we call it as gene flow. Very simple. Flow of genes from one population to another, we call it as gene flow. What is genetic drift? Again, very easy. When the gene flow happens multiple times, we call it as genetic drift. There is a drift in genes. The gene flow is happening again and again, multiple times. We call it as the genetic drift. These are the 3 points which you're supposed to cover under speciation. Next, we'll go to the last important question from this chapter, that is, what are the 2 different types of traits? Now, you have to be very careful with this, because if the question is from heredity part, then the 2 traits are dominant and recessive trait. That is totally different. What we have now is nothing but the acquired trait and the inherited trait. So the 2 types of traits are, trait is nothing but character. So we have 2 types of character, students. One is the inherited character and the second one is nothing but acquired character. What is inherited character? In the name itself we understand. The character that is inherited from the parents to the offspring. From parents we get it at the birth. That we call it as inherited character. For example, maybe the structure of your nose, maybe the structure of your ears, maybe the shape of your face, maybe the color of your hair, the natural hair color. All these are coming from your parents and these are called as inherited characters. What are acquired characters, students? The characters that we obtain during our lifetime because of the experiences that we have. These characters are called as acquired characters. For example, you may not know how to dance at birth, but you go to dance classes and you start learning dance, you become a dancer. Dancing is your character, but did you get it at birth? No. You acquired it in your life during your experience. So such characters are called as acquired characters. When such question comes, you explain what is inherited, what is acquired character, give an example for each and you're good to go. So with that, we have covered all the questions, important ones from Heredity and Evolution chapter, students. I hope your mind is now refreshed with this chapter. You know what to answer, how to answer, and we shall end here for today's session. Thank you, students. See you in the next session.